This video is on blood tests. We'll discuss what's in your blood, what the tests look for, and what the results might mean. Let's start with the three main cell types in blood. Red blood cells, also called erythrocytes, white blood cells, also called leukocytes, and platelets. Red blood cells carry oxygen throughout the body. White blood cells are involved in immune response, and platelets are involved in blood clotting. So if you cut yourself, you stop bleeding in part because of platelets. Now in the clinic, if you have blood taken, these cell types can be separated. If you just put blood in a tube and wait, the red blood cells will settle to the bottom of the tube. How quickly the cells settle can be measured, and this is called the erythrocyte sedimentation rate, ESR, or just sed rate. Increased ESR can indicate inflammation, which may be related to cancer, infection, or an inflammatory disease. However, ESR can also be elevated in normal, healthy individuals, so results must be considered in your own medical context. If you measure the size of that bottom fraction, divide that by the total amount of blood that went in, and make it a percent, you get the hematocrit, or HCT. In other words, hematocrit is the percent of your blood that's composed of red blood cells. The greater your hematocrit, the more of your blood volume is taken up by red blood cells. Another way to quantify how much of your blood is red blood cells is to use a microscope to look at the cells in blood. It might look something like this, with lots of red blood cells, some platelets, and a few white blood cells. You can count the number of red blood cells in a certain volume of blood to get a red blood cell count. A decrease in the red blood cell count, which might look like this, is called anemia. Anything that decreases the red blood cell count is a cause of anemia. Thus, you can imagine there are a very large number of different causes of anemia. One way to distinguish the cause of a particular case of anemia is to count cells called reticulocytes. These are immature red blood cells that have just entered the bloodstream. High levels of reticulocytes suggest the body is trying hard to replace the missing blood cells, which is what the body should do. Low levels suggest the body is having trouble producing red blood cells, so the anemia may be caused by a problem with production. Causes of anemia can also be distinguished by the size of red blood cells. In this image, let's say the red blood cells are normal sized, in which case it'd be called normocytic anemia. If the red cells are smaller than usual, it'd be microcytic anemia, and if the red cells are larger, it'd be macrocytic. Red blood cell size is reported on blood tests as mean cell volume, or MCV. It's also possible to have a wide range of sizes in red blood cells in anemia. The degree of variability in size is represented by the red cell distribution width, or RDW. A high RDW means the red cells are very different in size. Since red blood cells carry oxygen to tissues, and oxygen helps tissues to use fuels like sugar to generate energy, low levels of red blood cells, like in anemia, can result in symptoms of low energy. These are symptoms like fatigue and weakness. Other common symptoms of anemia are paleness, since red blood cells help to give our skin color, and shortness of breath, as the body tries to get more oxygen into itself. Now, sometimes the problem lies not in how many red blood cells there are, but rather how much is inside the red blood cells. Red blood cells are basically bags full of a protein called hemoglobin. Hemoglobin binds oxygen. The more hemoglobin in a cell, the more oxygen that cell can carry. You can think of red blood cells like delivery trucks, filled with hemoglobin boxes, with each hemoglobin box holding some number of oxygen molecules. If you don't have enough hemoglobin, or your hemoglobin is defective, not as much oxygen can be carried by your red blood cells. This can lead to the same symptoms as anemia. The amount of hemoglobin in blood, after the red cells are popped open in the lab, can be measured and may show up on test results as HGB. You may also see MCH and MCHC on blood tests. These have to do with hemoglobin levels, but tend to be less informative than the other measures I've talked about. That's all we'll say about red blood cells. Let's go back to the other cell types. Going back to our tube of separated blood, the white blood cells and platelets form a layer on top of the red cells. There's not nearly as much white blood cell and platelet as there is red blood cell, so this layer is quite small. Rather than trying to measure this little layer, the number of white blood cells and platelets are just counted under a microscope, like we can do for red cells. These counts, plus the measures we talked about for red blood cells, make up a complete blood count, or CBC. If counts of platelets are low, this can result in easy bruising, nosebleeds, heavy periods, more bleeding after the dentist, or other abnormal bleeding. Note, though, that there are many other factors involved in stopping bleeding, so more bleeding isn't always a platelet problem. If white blood cell counts are low, there may be an increased susceptibility to infection, but this effect is often too small to be noticed. Now, there are actually many different types of white blood cells. The different types have different functions, though all are still related to immunity, and the different cell types can be distinguished under a microscope. A count of how many there are of each white blood cell count in a certain volume of blood gives us a white blood cell differential, often just called a differential. We'll have a quick look at the types of white blood cells that are typically counted. Most white blood cells in an adult, 60 to 70%, are neutrophils, which are involved in defense against bacteria. Basophils are the rarest and are involved in allergic responses. Eosinophils are involved in defense against parasites. Monocytes can enter tissues and become macrophages, which can consume dead or dying cells or infecting cells. Lymphocytes include both B and T cells. B cells are involved in antibody production, and T cells help defend against viruses and cancer cells. Identifying which types of white blood cells are most effective can help suggest why a white blood cell count is abnormal. For this purpose, a particular type of neutrophil, band neutrophils or band cells, may also be counted. Band cells are just immature neutrophils. An increased number of band cells may be a sign that the body is trying to pump out a lot of new neutrophils, which may occur normally in response to infection or be a sign of certain blood cancers. So we've talked about the cells. Let's move on to the rest of the volume of blood, which is a fluid called plasma. Plasma is a bit like the broth of your chicken noodle soup, with the cells being the noodles. Plasma contains vitamins, minerals, hormones, carbon dioxide, and proteins, one of which is fibrinogen, a protein involved in blood clotting. If you've heard the term blood serum, serum is the same as plasma, except that it doesn't have fibrinogen. When a lab isolates blood serum, they allow the blood to clot, so the fibrinogen sticks together and can be separated out. Many of the proteins in plasma can be measured. For instance, lactate dehydrogenase, immunoglobulins, beta-2 microglobulin, and albumin can all be measured to indicate disease severity. About 60% of all the plasma proteins are just albumin, so I really should show it like this. The function of albumin is to keep the solute concentration of plasma high enough that enough water remains in the blood vessels. Even though it ends up in the blood, albumin is actually made in the liver. Measuring albumin can suggest impacts of disease elsewhere in the body on the liver. Many diseases, including blood cancers, can result in lower liver function and less albumin production. I've also mentioned lactate dehydrogenase is measured as a marker of disease severity. Lactate dehydrogenase is involved in cell metabolism, and so is normally found inside cells, not free in the plasma. Elevated lactate dehydrogenase in the plasma can suggest that cells somewhere in the body are breaking open, which may be the result of many different disease processes. Of course, blood sugar, cholesterol, and different types of fats are also commonly measured in blood, but we won't discuss those in this video. In summary, we've covered the cell types and plasma of blood, how they're measured, and what functions they perform. For more information about where blood cells come from and how they relate to blood cancers, please see my other blood cancer clips.